Right, what a great audience. Good to see so many faces keen to get out there and do more for arboriculture. My name's Mathew Daniel, I'm from Tree Preservation Australia. So what we focus on is soil health. I've been really interested I've been an arborist for 20 years, but about 10 years ago I got really interested in managing trees sort of more holistically. So looking at, what's happening, overall, including a lot of the stuff in the root system. I went to Boston. I was invited to Boston Tree Preservation, and over there I studied bioremediation and got exposed to the industry over there. Learned a lot about what's happening in the soil, and I think probably in the last 10 years I've started to realise that there's probably a lot more that we don't know. I think the more that I learn about soil, the more that I realise I don't really understand much at all. It's extremely complex, and I think we're getting to the stage now where all we really know how to do is ask the right questions, which sounds bizarre, but it's allowing us to sort of understand things more and remediate tree health. So TPA is made up of myself and Gerhard Grasser. Gerhard Grasser's been around for a long time in agriculture. He couldn't make it today. So we work on holistic plant healthcare management, consultation training, specialising in biologically driven maintenance and recovery of stressed and declining trees and their surrounds. All right, so we're going to run through a case study today towards the end on a tree that we've worked on. And what I intend to do is to try and see if I can link the subject of habitat in with sort of soil management and get us to sort of start to acknowledge a little bit more about what's happening below the ground, because the soil biology is really important to the overall habitat. Now what I also want to do is I want to try and see if we can link ecology and not just talk about habitat. So biodiversity and ecology is really important when we're talking about habitat. So I've taken the lead here from Shane with Talking Animals, if you all saw the flyer that went out. So to start off, ecology, the study and relationship between living organisms with one another and their physical environment or habitat. So it encompasses two factors, which is biotic, the living, and abiotic, the non-living. So you can see there we've got a little microarthropod talking to a little mineral particle. The microarthropod's biotic, it's a living organism, and the mineral particle is abiotic, it's non-living, and you can see there a little nematode. I'm not sure if it's a bacterial feeder or a predator. It's quite big, so it's possibly a predator. So when we're talking about ecology, the first thing we've got to understand is that we're talking about the two factors there that are really important to include, the biotic and abiotic parts of an ecosystem. Interdependence is a really important factor when we're thinking about habitat and ecology. So the survival of a species is dependent on other living organisms and non-living elements. This is called interdependence. So you can see there we've got a little sparrowhawk that I found down at Merry Creek, out in the open, not in a tree. I've seen him a couple of mornings in a row walking the dog, which is, I think, maybe interesting if anyone out there knows a little bit more about sparrowhawks. But that interdependence is really important. So you've got a predatory bird there, and if you're thinking about food chain, that predator is part of a food chain, and he's looking for smaller birds and things like that to eat. I think they also eat worms and things, but if you're thinking about the food chain, the worms are really important. And if anyone was in the Soil Secret seminar, can anyone tell me what worms eat? No?
Bacteria.
Yep. Bacteria, fungi, protozoa and nematodes. So worms are primarily feeding on all those lower order organisms. So if you're not seeing worms, it means that you don't have all those lower order organisms in the equation. So worms are a good indicator that you've got that ecosystem functioning, because you've got all those lower level order organisms in there. The largest known ecological system is the planet Earth, which is a global biosphere. And the biosphere is everything from the deepest oceans right the way up to the tops of Mount Everest. So and we're part of that biosphere as well. Okay, so the components of habitat inside an urban forest ecosystem, we've got our abiotic parts, the non-living parts. So we've got precipitation, water, falling leaves, branches, flowers, sunlight, soluble mineral nutrients, oxygen, carbon dioxide, soil pH and soil temperature. And then the biotic parts of that ecosystem is our tree, our primary and secondary consumers, so like our birds and animals, and then our soil microbiology, which decomposes, and microbiology can also be part of the consuming pool. Habitat for microbes inside an ecosystem. So we've got our... Oh, jumped the gun there a little bit. So we've got our habitat, so we've got our root zone there. Oh, now this is our case study tree. So this is the Church and Dendy Street Gladicalyx that we started working on last year. So we've got our habitat is our root zone, so a place where an organism or population of organisms can live. And then we're looking at a close-up version of this area here. This is post-PHC program, so this is after we did our biological remediations. And the niche is the primary decomposers, so all the ways that an organism interacts with everything else in the ecosystem, the role or the job of the species in that organism. So in this situation we've got our saprophytic fungi, and their role or job is to break down this organic material, the carbon, and recycle the nutrients and enable disease suppression and all that sort of stuff. Really important part. So the trees, habitat inside an ecosystem. So the habitat is the urban forest, a place where an organism or population of organisms can live. And you can see from that picture that increasingly our ecosystems are really suffering in the urban forest. Lots of pollution, and we have a really big impact on biodiversity on the urban forest. And the niche of the trees inside that urban forest is photosynthesis. So all the ways an organism interacts with everything else in the ecosystem, the role or job of that species in the ecosystem. So it's really important that we think about that. Primary productivities are really... I'm just going to sort of go over some important points in ecology that I think we should think about to sort of tie today all together. So primary productivity, the measurement or the rate at which organisms convert light energy or inorganic chemical energy into biomass. The producers in an urban forest are the organisms using energy, sunlight, making photosynthesis, or organic chemical energy, nitrogen, and weaving matter to make life. So our two main primary producers in an urban forest are going to be our trees and plants and the microorganisms. Now, not all the microorganisms are going to be primary producers, but if we think of azotobacter, like nitrogen-fixing bacteria that can take chemical energy out of the atmosphere, nitrogen out of the atmosphere, and use that, and it's basically the engine room of good ecology and biodiversity, because without a lot of primary productivity, we're not going to be able to increase biodiversity or really have a benefit to a good ecological system. So primary productivity is measured in grams of carbon per metre square per year, and this is basically carbon sequestration. And primary productivity is crucial to biodiversity. Okay, so we'll start talking about... I've missed a couple of points there, and if we've got any ecologists in the audience, you'll know that I've missed some really important points there. But we don't have all day to talk about ecology, so we'll sort of move on and start getting into it. So what's important with us as arborists is the soil food web, part of an ecological and healthy biodiverse system. So this is our soil food web. We've got our primary producers here, the plants taking energy from the sun, photosynthesising, and being that engine room for the whole ecosystem. Now, if you move up, you've got your fungi, your bacteria, your nematodes, and your protozoa, and there's a lot of nutrient cycling and disease suppression that happens in all this area. One thing we don't see in this picture is worms, and worms will sort of be in this sort of area through here, and because worms are feeding on all of our bacteria, fungi, protozoa, and nematodes, and this is a good indication of what is happening out there with good ecology and what we need to think about when we're thinking about habitat. You can see we've got our birds and our animals, here, and without all of these other things, we can't provide good habitat for these guys. And you can take one of these things out of the equation, and it really does affect how a soil food web works. We measure the soil food web. We've been measuring the soil food web for 10 years now. Gerhard's been doing it for about 15 years. And we find all the time really big problems with these guys, you know, at the start of the food chain. And when that happens, it affects, you know, everything else, you know, all along the way. One thing we see in the urban forest all the time is that we, especially our mycorrhizal fungi, we don't see a lot of mycorrhizal fungi in the urban forest. And very rarely do we see good amounts of just disease suppressive fungi, good saprophytic growth, and that's because we're affecting that primary productivity, and we're not thinking about carbon sequestration. We're really lowering biodiversity, and we've even had situations where we've looked at trees and we've found that there's just no bacteria, and if you don't have those single-celled organisms in the very beginning, it affects everything else along the way. Okay, so the important factors of what the microbes are doing, microbial interactions with trees. So trees produce their own food in the form of photosynthesis. So the only things on Earth that actually produce their own food. Most things actually have to go out there and source food. So plants are an amazing thing, the way they can do that. Most of the energy so plants only use 1% or 1 to 2% of the energy that comes from the sun, and they use that in photosynthesis. A lot of that other sunlight gets reflected off back out into the atmosphere. So there's a huge deficit there. And the other side of it is that they don't actually use all of that energy that they make through photosynthesis to grow their leaves and the stems. They store a bit, but a lot of it gets actually dumped out into the soil profile, and that's in the form of exudates. And they do this so that they can basically communicate and talk with the microorganisms. They don't talk verbally like we do. They do this through the chemical exudates, and it's a recipe. It tells the microbes what the plant requires, what to do at different times of the year, and it can change through the day, and it can change through the season, and it's a really important part of how trees function. And they do this because they want to have root protection, both physical and enzymatic. They want nutrients, and they want the microbes to help them build new soil and to navigate through the soil profile. So things like mycorrhiza, if we're a group of arborists, mycorrhiza is a really important part of the soil food web that we should all remember from school. And one of the big things that mycorrhiza do for trees is to protect the trees. It's basically a force field that stops anything from attacking the tree. So if you think about a balloon, I can push my fingers inside a balloon, and I don't rupture the balloon. And that's basically how mycorrhiza work. They can get in between the cell wall without breaking the cell wall, and then they encapsulate the cortical cell wall and stop diseases from penetrating it. They're also extremely aggressive, so they can get out there and actively attack diseases and consume diseases. So this is a little bit of an indication of what's happening there. We can see nice big root to shoot ratio, which is what we should be trying to achieve out there in the urban forest. If anyone's done any sort of root excavation, I think you'll find a lot of the time we find a really small root to shoot ratio. And you can see here that the worms are feeding on all the lower order microorganisms, organic matter and debris. The CDN ratio is really important as an extra food source for these guys. The trees photosynthesising, producing exudates, dumping it out into the soil profile to feed these guys and to communicate with them. We move on to soil biological succession drives plant succession. So succession is a really important thing for us to understand too when we're talking about habitat, because essentially that's what we need to do. We're not going to be able to produce habitat, in situations like this. We need to have a healthy ecosystem and a lot of biodiversity to be able to sustain habitat and good healthy ecosystems. Now it's the biology that drives soil biological succession. So you can see here, in the beginning we've got sort of parent material, and we've got really low amounts of bacteria and no fungi. And then as you move forward up here, we're able to sustain more fungi and more bacterial growth, and that in turn supports a healthy forest ecosystem. Now, from our measuring that we do in the urban forest, quite often, you know, we're dealing with soil profiles like this. We're dealing with, you know, soil biology that's trying to support an ecosystem at this level, and that's why we're having a hard time at it. That's why you see a lot of decline in the urban forest, because it's just you just don't have the engine room there to be able to support a healthy ecosystem. So, you know, the biological succession is a really important part of promoting a healthy ecosystem and biodiversity. Now, these are really good interpretations of what healthy, of the sizes of the pools of things that we need in a healthy system. So if we start up the top here, we can see the root to shoot ratio. We've got a really nice big amount of root mass supporting the above ground mass. We've got a good, if we're talking about trees in this situation, we've got a good pool of mycorrhizal fungi, good pool of saprophytic fungi. Bacteria is being kept in check by all the flagellates, and then, you know, all of this, you know, functioning can support all of our higher level predators. And, you know, if we change that box from higher level predators to, you know, birds and things like that, you can, you know, that's the sort of, that's what we're trying to achieve. But we're not going to be able to achieve good habitat and biodiversity, you know, ecological systems without all of these other guys as well. They're all important. What else can we can see that up the top here we've got a small block of what are those? Plant-feeding nematodes. So, yeah, that's a healthy system. When you look at an unhealthy system, you can see that we've got a large pool there of plant-feeding nematodes. We don't have any mycorrhizal fungi. We've got a little bit of saprophytic fungi, but we've also got a lower pool of organic material, you know, feeding that saprophytic fungi. Bacteria is way out of balance, and, you know, I can see in this situation, we see this a lot in the urban forest when you look at the data, because this is basically an interpretation of the data. Quite often you see in compacted soils, you see, you know, a large pool of ciliates. Whenever you see a large pool of ciliates, you always tend to have really high levels of compaction. Now, you've also got, you know, we're not supporting the higher level predators here, so if we're thinking about habitat and looking at, you know, this data, the system's not functioning properly to be able to support, you know, a lot of predators and a healthy ecosystem. So, and we can change this. It's quite easy to change that. We'll get to that down the track. So yeah, you can see, and you can see there it also affects the tree's root to shoot ratio. So we've got to address those sort of issues. So healthy system and unhealthy system, which is what we see all the time out there. Okay, so get on to the case study now. So this is the Church and Dendy Street Gladocalyx that we started working on in December last year. On the corner there of Church and Dendy, really high tree decline. It's not a very strong biodiverse system going on there. So outline of the remediation program, so that we started off with collecting soil samples of analytical data. So we look at soil chemistry, soil biology and pathology. Then we look at collecting benchmark physical site characteristics. So we measure compaction, leaf temperature, soil moisture, soil pH, a visual soil assessment. We do that as well as a visual tree assessment. Then we interpret, we get all the data back from the lab, and then we interpret that data and then design a program to remediate all of those issues. And then we retest afterwards to see what's happened there between start and finish. And then it takes about 2 to 3 years to effectively fix a lot of these things when they're in bad shape. We'll get to that data. So this is, yes, still in around December. We had severe tree decline. We found canker lesions. We found a duplex root system, high soil compaction, historic TPZ impacts. And I've got another picture there that will show where that is. Now, what's interesting is that the mulch layer had been there for quite some time. It's quite a sandy soil in this spot, and when we dug our soil profile pit, you could see that the mulch had been there for years, and there was a clear layer between the soil surface and there was about 2 or 3 different sort of mulch layers, but they just weren't breaking down. And when we looked at the data, you could see that just laying mulch down doesn't necessarily always bring in and activate a healthy ecosystem, because if you don't have the soil biology there in the first place, you're not feeding it with all that carbon that you're bringing in. Mulch, you'll quite often bring some other organisms in, but quite often we see this a lot, that the mulch will just sit there inert for quite some time. So it's not improving an ecosystem. It's not supplying good biodiversity, and in turn, providing habitat for other organisms. So yeah, it was said there in the bottom there, we've got no evidence of food web activity. We didn't see any signs of birds scratching around in the mulch or anything like that. Quite often our clients get a bit upset with some of the projects, because we tend to find that after we inoculate the site with soil biology and you activate that soil food web and you start building an ecosystem, the birds come in, because we've got worms starting to feed on all those other lower order organisms. Some other things that we found on the site. So we've got historic TPZ impact. So we found that there's an old Telstra pit, I think it was about here somewhere, and it looks like they've just cut a great big trench down here, and then there's another pit over here. I think it was in the days of telecom, because it looks like there's a fair bit of old root decay there, but just incredible that type of invasive works was done around a heritage listed tree. The other things we found, we got a big limb was cut off the tree after we did the initial testing. So this is a couple of months. This must be about January or something like that. And I think that's around when this big limb was cut off. It was a big stem bark ridge cut. The limb had declined, but I think we've got to be careful making big cuts like that on old trees, especially when they're in such a state of decline. And we came along one day and found that one of the residents was going to have a carriage sale down at 149 Church Street, and they used a 4-inch nail. It was to put that sign up. It was just incredible on a tree that was really sick. It was this tree was in critical condition. It was touch and go to try and see if we could save it or not. Okay, so how do we go about fixing the soil health? We call it the new NPK. So can anyone remember what NPK used to stand for when we went to school? Come on. Nitrogen, potassium, phosphorus. Yep. That was a really narrow way of looking at what's really important when you're dealing with soils. The more that we understand now is that you've got 3 really important components of soil, and they are nutrient, physical structure and key biology. And you take one of those things out of the equation, or you have a major impact on one of those things, you're going to affect the whole way that soils function and how they support good tree health, and in turn, habitat for plants and animals. So the first part of the new NPK is nutrient. So this is before and after. So you can see there we've got toxic levels of nutrient there, and excessive, and deficient, and highly deficient. That's just Gerhard is the expert when it comes to chemistry, but you can expect that plants are going to struggle when you've got soil conditions like that. It's completely out of balance, and it's just not going to support good healthy tree growth. Now after 4 months you can see that everything's sort of starting to balance out there. It's not perfect yet by far, but it's starting to balance out, and there are some interesting things in there. I don't have time to go into them all, but the total sulphur is we're starting to see that as a real issue in the urban forest, and that'll be affecting our mycorrhizal colonisation as well. So we'll get to the data on the mycorrhiza, but I think that's why we had a bit of trouble with the mycorrhiza there. getting that to grow. The other side of it is the tree was in decline. So if you know anything about mycorrhiza only inoculate active, healthy root mass. They won't. If you put mycorrhiza out there on a dead or dying tree, it's not going to work very well. So we can see that in the data. So if anyone's interested in the chemistry there's some interesting stuff there. So the physical side of it. The really interesting part here is the compaction. Oh. So when we started, we're looking at 600 to 700 psi at 40 centimetres. So there was a clear subsurface soil compaction layer. And that's partly indicating where we had the duplex root system. But that's a really unhealthy compaction layer. Healthy root mass will only grow in about 300 psi, so anything higher than 300 psi is really going to limit good healthy root growth. And you can see there in just a few months, and it's the biology that's doing that. We didn't do any air knife work or anything like that. It's just biology that's doing that. It's just the soil microbes that are doing that, and that's quite amazing. What I found interesting is that the compaction in the first 10 centimetres really reduced, and you could feel it. When you walked on site afterwards, you could almost feel the biological activity because it was sort of spongy and it was when we first started, there was a real hydrophobic layer. Like the area just wasn't absorbing water, and then as you start to sort of ameliorate it and fix the soil biology and balance it all out, it's able to absorb water. So the compaction layer is a main one. pH didn't really change, but we call it pH indication because we use a pH probe, and the way they work, they're not very accurate. They're not as accurate as what the laboratory assessments are. But we use the probe because quite often it's a good indication of issues in the chemistry that we can see really quickly. We quite often catch gardeners out. Garden managers out on site when we ask them if they fertilise with any chemical fertilisers, and they always say no, and then we find really low pH, and then you find excess fertilisers in the ground when we start digging around. The leaf temperature was interesting as well. That's a new leaf, so that should be a little bit lower, that temperature on a new leaf. And the older leaves were at ambient temperature, which is telling us that the tree was not drawing moisture very well. That's about it of interesting information there. Oh, the soil moisture, that was interesting too. So that that balanced out. After a while, we ended up with a more balanced reading, average reading from the site. Where we started, we had sort of different readings around the area. So then this is the interesting stuff that I get excited about. So the new NPK, the key biology component of it. So when we started, we've got total fungi. Total fungi at the top was sort of in range, and then we moved those to above range. The active fungi were below range, and then they moved up into in range. Now the interesting stuff is the bacterial levels. The bacteria are your single-celled organisms that are the very beginning of everything else feeding on. So you need to be able to support all the protozoa and the nematodes. You need to activate that bacteria, and that happened. Another interesting thing is the ciliates. Now, if we think back at the compaction layer, the ciliates decreased rapidly. So that's lining up with the compaction layer. So wherever you see high compaction, you see high levels of ciliates as well. We see that consistently in the work that we do. And that ciliates are also an indicator of anaerobic conditions. Now, what's interesting with this site is because the tree was in such a state of decline, we didn't use lots and lots of mycorrhiza because it's quite expensive to apply mycorrhiza, but we did do it, and the mycorrhiza went backwards, but the tree health was going backwards as well. So there's no point putting mycorrhiza out on sick trees. You need to work on all the lower order organisms before you start thinking about things like mycorrhiza, and also sometimes with your different beneficial fungi as well. And if we put those into a percentage, you can sort of see there quite quickly what happened. It's also interesting too when you start seeing numbers like this. It just goes to show how important the soil biology realm is and its capacity to do quite incredible things. That's a big number, and that's a big number in 4 months to increase by. And this is the sort of stuff that when you look at soil biology, you realise how important that realm is to biodiversity and habitat and good, strong ecological systems. This is how we actually, generate the power for a healthy ecosystem. It's the storage, it's the energy production. It really starts in this area. And without this functioning, you're not going to be able to build on, that healthy ecosystem. So you can see there we've got, active bacteria have increased by 113%. And if we've got good bacteria, then we're going to be able to, the things that feed on the bacteria are going to be able to be supported. Now you can see there that the ciliates have decreased by 80%, and our compaction, it sort of mirrors that. So our compaction layer had really reduced, and we had a declining tree, so it makes sense that we've got dying roots. So you're not, your mycorrhiza is going to go backwards as well. Active fungi going down by 10%. Could be the time of the year that we had, but that just shows quickly what's happening there. So in summary, we collected data. We looked at the new NPK, so the nutrient, the physical structure, and the key biology. And we improved that to try and increase the microbial habitat and have a positive impact on an ecosystem. So a shift in soil health based on better microbiology can be achieved to support healthy tree function, provide habitat and improve an ecosystem. So we can see when we first started there, you can see that clear, it's not a very good picture, but there was a clear layer of between this mulch layer and the soil layer. The mulch had been there for years and years and years and just wasn't breaking down, and that was because the biology wasn't intact, and we also had toxic chemistry that's going to inhibit that microbial activity. We had some sort of unhealthy root growth in the mulch, and then afterwards you can see that we're starting to support an ecosystem or improve an ecosystem by bringing in the biology. That picture there is a sort of a sandwich. It's a layer of hyphal growth that's been lifted up and turned back onto the top layer of the mulch. You can see here a lot really active growth, all this light grey fuzzy stuff, and then the active mycelium. So a healthy habitat and ecology includes single-celled organisms, and we can't forget that. It's important to remind ourselves that to provide a habitat and improve an ecosystem, the smallest of organisms, the soil microbiology, cannot be ignored. That's me.
We've got a little bit of time for a few questions. If anyone has a question?
Shoot some questions. We can go back to the data.
Mr. Kenyon.
What happened with it? Should we go?
It's not good. Yeah, we got in too late. Yeah, so it's. I think there's a fine line there to know when to get in, and that we're working on that research at the moment. Actually, trying to figure out plant indicators, not just visual indicators, some actual analytical data to be able to measure to know when you can have a successful outcome. But it was a significant tree, so we were asked to try and throw what we could at it in a short amount of time. But in 4 months, you can see that was just a 4-month bracket of soil remediation. And if the tree hadn't have been it hadn't been in that state of decline, if we got in there, maybe 4 months before then, we would have had a much better shot at trying to get it to balance out. Yeah. But it's a good tree for a habitat site. So that's probably my point in a way, is that, something should be done with that tree now. You know, it should be retained in the landscape, and it's a good candidate for habitating, and to keep there as a reminder that we should get in before, you know, you get to that level of decline, because it's clear that you can turn a tree around if you've got issues with soil health. You just need to get in there at the right time. Yeah, mate?
I saw a lot of comparisons between what it was like before and what it was like after, but I didn't see much of what you were applying to start to change these things.
Okay, so we applied, so looking at the data, so the first things we start on is the single-celled organisms. So we start on bacteria. So we actually grow bacteria and then put bacteria out and then feed that bacteria. And then we bring protozoa in and then feed the protozoa and stimulate.
You're building it from scratch?
Yeah, starting off with the single-celled organisms and then moving on. We put the mycorrhiza out. We didn't put the mycorrhiza out in the early stages. We put that out sort of about halfway through the program. And the thing about mycorrhiza is you're putting them in. You're not putting them in an active form. You're putting mycorrhiza out in a spore form, and you have to get that spore to an active root. If you don't get it to the cortical cell wall, it's a complete waste of time and money. So that was a hard one to sort of balance out. Well, when should we be putting out lots of mycorrhiza? Because there's no point in putting out lots of mycorrhiza too in compacted soil. So you've got to work on those lower level order organisms first before you start working on the higher order organisms. So yeah, so to answer your question, we started off with inoculations of bacteria and protozoa, and then we moved off into more balanced sort of bacteria and fungal inoculants, and then feed sources. We didn't put any extra mulch out. One thing I wasn't able to show you because I didn't benchmark the data, but the level of mulch dropped dramatically. So when we started, the mulch layer was about that deep, and when we finished, the mulch layer had halved. So all those microbes had broken down all that cellulose and carbon and recycled it, and yeah.
What time period was that over?
4 months. Yeah. Well, it's not quite. It wasn't 4 months of treatments. It was 4 months between testing and retesting. So it was probably only a couple of months in actual inoculations, because we had Christmas as well. So I tried to get out there as quickly after Christmas. Any other questions about data? Yep.
Yeah, I was just wondering. You're dealing with a very reduced root spread area.
Yeah.
How did you go getting those inoculants down into like just a structural root zone area where there's not a lot of feeder roots?
Yep.
And so it makes it a little bit difficult for all those inputs to actually work in the feeder root zone area when you're dealing with the structural.
Yes. Yeah. So we had 2 layers of root mass. We had this older subsurface layer, and then we had all this feeder root mass. Trying to source stuff in the old mulch layer. So what we started off at first, because we had a compaction layer there, we did some soil probing. So it's just our soil probe's about 500 deep, and so we punched down, down through that mulch layer, and sort of, probably punched down into about that much of that subsurface soil layer, and then it was drenches after that. So it's a little bit of probing and then sort of drenches. And then towards the end, you don't need to probe anymore because the compaction layer is reduced. We were watching that compaction layer. So every time I do a treatment, I collect all that physical data. And the compaction layer was reducing really quickly. And so towards the end, we were able to in that area there, I was able to put out about 1,500 litres of inoculant really easily. Where when we started, the drenches it was only absorbing about 500 litres. And so that's a huge increase, for that area to be able to absorb an extra 1,000 litres in a couple of months is really incredible. And then when you look at that data, when you look at a 169,000% increase in one protozoan group, it's just huge numbers. And we'd like to see those numbers increase across, throughout the board, but it takes a little bit more time to be able to do that, and better tree health as well.
And is your expectation over time that all that, because mycorrhiza can stretch for quite some distance, it'll seep down, maybe go under the road verge and
No, I think what happens with this, I think the spores, you water the spores in, and they'll get caught up in some organic particle, and they'll stay there until a root passes by. And as soon as you get a root passing by, then that spore will activate and start growing. And we've got other sites where you can actually see, mycorrhiza is one of those smaller, lower level organisms that you can actually see after a bit of time. We've got some other. I've got some photos, I think, on these slides where you can see you can actually see with the naked eye. You can see the mycorrhizal activity, but it's very difficult. And see, in a site like that too, is that because we had so much, root invasion in that area, we were trying to limit how much impact we had on the area as well. And we were trying to see if we could, activate some, fibrous root mass first, just to sort of get the tree to start functioning before we did anything more. But it's a hard balancing act sometimes, because you need to do a certain element of investigation before you start making calls on what to do. But quite often in that situation, you've got very limited root mass that you're working with in the first place. So, yeah.
Harsh environment.
That's right. Yeah. And it's interesting from the chemistry data, when you look at that, you can, those high levels of sulphur, that's where we're seeing that really consistently in the urban forest, and it's a real issue. If we're getting sulphur levels, absorbing into the soil, it's going to make it really difficult to be able to remediate, tree health because of that. And I think it is an urban forest thing, because when you look at the agricultural data that we've got, we don't see those levels consistently. And that level too, I think the high sulphur levels it's starting to be a bit of a consistent theme. We do a lot of work on cypress canker, and there seems to be a bit of a consistent theme between the high levels of sulphur and cypress canker. So yeah.
It makes sense with all the, like Yarra Valley, with all the inputs and fertiliser and what's happening.
Yeah.
Thanks, mate. We've got one up in the centre aisle. Yeah.
G'day. Just a quick one on, if you had the luxury of time, let's say you have a property with a large number of trees on it, you wanted to address them as like a project approach, is it a really good time of year? Like, is it probably, you do some of that prep just before the spring growth, or perhaps an autumn revamp if you're doing late summer, and then the roots sort of grow through and stuff? So like
we find with the sort of native trees, evergreens and stuff like that, winter time's a good time to take advantage of the higher levels of moisture, and also sort of those extreme temperatures as well, because the extreme temperatures really have an impact on the soil biology. So yeah, it depends on the types of plant material that you're working on. When you're talking about deciduous plants, before you've even seen any sort of bud swell, you've got a huge amount of activity in the root system. So before we see any sort of indication that the plant's changing and coming out of a season, there's a huge amount of activity in the root zone, in the rhizosphere with the soil biology. So that's a really key time to actually get in and do some things. It depends on what your issues are, whether you're going to look at needing to inoculate or feed. Sometimes we look at, so you've got two things with biology. You've got your total numbers of soil microbes, and then you've got your activity. Because microbes are going through a constant state of flux, there's things waking up and doing their job, and things going to sleep and all of that sort of stuff. There's a difference between putting microbes back out that are missing and feeding the ones that are there, or the ones that are about to wake up. So you think about what type of plant material you're working with, and what time of year you're working with, and then the conditions that you're working in. So your physical parameters and your abiotic parameters, and you sort of match those things to what you're trying to achieve. So there's no silver bullet approach to one thing. You've got to. That's why it's that NPK approach, your nutrient, physical structure, and key biology. And what's missing in that scenario is your plant material. You've got to match those three things to whatever you're dealing with. You're going to do something different if you're dealing with a turf situation, a monocot to a dicot, and then to a deciduous plant to an evergreen plant.
We've got time for probably one quick question over here.
Yep.
I'm just wondering, was
Yeah, I think so. I think it was a safety issue, but I think it was a poor arboricultural call. I think you could have just reduced the weight, but I think to go back to an SBR cut was just a bit harsh, especially on a tree like that was so sick. And I don't know, it's hard to tell if that was a contributing factor. The tree started, after that happened, the tree started producing a lot of anthocyanins, so it went really purple straight after that happened. So I don't know, it's hard to say, you know, make the linkage there. It was just a whole lot. It was a bit of a hard case. You've got people nailing nails in the trees, big branches getting cut off it. You've got, a 20-year-old, impact on the TPs there, the critical root zone, and then you've got toxic chemistry, and then you've got inadequate soil biology, and a lot of physical problems like the tree. The tree is really struggling, to be able to survive and until you address some of those issues. Yeah. And we see that all the time in the urban forest. You have a look around there and start measuring some of those things, and it makes you wonder how some trees can survive, and it makes it obvious how a lot of trees are declining like they are.
Okay, thank you very much, Matt.